Assalamualaikum dear student. Today we are going to discuss few more detail about the nucleic acid. The history of nucleic acid. The DNA was first isolated and characterized by Frederick Mesher in 1868. Avery, McLeod and McCarthy proved that the DNA was a genetic material. Avery and his colleague found that the DNA extracted from the virulent strain of the bacteria Streptococcus pneumoniae injected into a non-virulent strain of the some bacterium transformed the non-virulent strain into the virulent strain. They concluded that the DNA from the virulent strain carried the genetic information for the virulence. In 1954, experiment by the Hershey and Chase in which they studied the infection of the bacterial strain by the virus bacteriophage was radioactively labeled DNA and protein. Any remaining doubt that the DNA not the protein carrier of the genetic information. Erwin Chargaff and his colleague in late 1940s proposed rule known as the Chargaff rule, which played an important role in the discovery of the double helix structure of the DNA. The base composition of the DNA varies from one species to the another. DNA specimen isolated from different tissues of the same species have the same base combination. The base combination of the DNA in a given species does not change with the organism age, nutritional state, and changing environment. In all the cellular DNA, the number of the residues is equal to the number of T residues, and the number of G residues is equal to the number of C residues. That is, the sum of purine is equal to the sum of pyrimidine. Rosalind and Franklin and Maurice Wilkin showed that the DNA produced characteristic X-ray diffraction pattern. In 1953, James Watson and Francis Crick postulated a 3D model of the DNA structure which consists of two helical DNA chains bound around the same axis form the right-handed double helix. Let's see what is the Chargaff rule. The base pair rule of the DNA is known as the Chargaff rule. What does that mean? The adenine, which always pair with the thymine, and the guanine always pair with the cytosine. Okay. Therefore, in a DNA strand, the percentage of adenine is equal to the percentage of thymine, and the percentage of cytosine is equal to the percentage of guanine. Example: if a particular DNA molecule is 30% adenine and then it is 30% thymine, which is equal to 60%. 30 plus 30 is equal to 60. And out of 100, what is left? It is the 40%. This means that the remaining 40% is that it means when you will have to divide the 40% to the two part, that is the molecule out of 40%, 20% is the cytosine and 20% is the guanine. I hope it's clear to you. Note, the Watson and Crick is refers to as a B form of the DNA. Over here, there is a diagram of a left-hand thumb rule and the right-hand thumb rule. Okay, because it shows how the helix is formed in the DNA structure. Let's take a look. Its features are: two long polynucleotide strands are bound around a central axis to form a right-handed double helix. The hydrophilic backbone. What was the hydrophilic backbone? Do you remember? It is the phosphate. Okay. Are on the outside of the double helix facing the surrounding water. Okay. The furanose ring of each deoxyribose is in the carbon number 2' endo conformation. The purine and the pyrimidine base of both the strands are stacked inside the double helix, with their hydrophobic and nearly planar ring structures very close to each other and other perpendicular to the long axis. The strands are anti-parallel. One strand is in the orientation of 5' to 3' and the other is 3' to 5' direction. Okay. I hope it's clear to you. Why? Because when we study the two strands of the DNA, we find that One strand has a polarity of 3' to 5' direction and the other has a polarity of 5' to 3' direction. Okay, let's see the double stranded. What is the DS? Is double stranded DNA structure. Okay, in this diagram you see these are the base pairs. They are inside, okay, base pairs, and these are the sugar phosphate backbone which is outside. This is the major group because it is a long gap, and it is a minor group. You can see the gap distance is so small. The axial rise is 3.4 angstrom. What is the helix diameter? Is 20 angstrom. One helical turn is 10.5 base pair per turn. What is the helical pitch? These are the helical pitch. In physics also you have studied what the helix or how the pitch is formed. Okay, when you have studied the wave. Okay, the same thing happens here. The helical pitch is 35.7 angstrom. Okay, let's see. The helical diagram is 20 angstrom. Helical rise per base is 3.8 angstrom. Helical pitch is 34 angstrom. What is the helix pitch? The distance along the axis per 360-degree turn. Okay, 10.5 is the base pair per helix turn in aqueous solution. Okay, always remember this value can only be possible if the DNA is placed in an aqueous solution. The strand interact with the hydrogen bond between the complementary bases. What are the complementary bases? That is AT and GC. Always remember it. Okay, the GC form 3 hydrogen bond with carbon. What are the number of hydrogen bond that is formed between the G and C? That is the 3. And what is the hydrogen number of hydrogen bond that is formed between A and T? That is the 2. So A form 2 hydrogen bond with T. Okay. Of the base pair relative to the helix axis as described by another 3 parameters. Okay. Base pair tilt, base pair roll, and propeller twist. Okay, this is a very simple diagram because as I mentioned what is the tilt, what is the roll, and what is the twist, you are unable to understand. So in this diagram you can see by the figure you can remember. If I say these terminology, always this diagram should be strike in your mind that how it should be look like that. Okay, in tilt the figure should be like this. When we are talking about the propeller twist, the figure should be like this. Okay, let's see what is the base pair tilt. It is a shift of the base pair short axis relative to the vertical helix axis. The tilt angle is measured by considering the angle made by the line drawn through the 2 hydrogen bonded bases relative to the line drawn perpendicular to the helix axis. Okay, note the tilt angle open in the direction of the phosphate backbone. If it is open in this side, what should be there? There should be a phosphate backbone. Okay, this is the base pair rule. What is the base pair rule? It is a shift of the base pair long axis relative to the vertical helix axis. Okay, what is the propeller twist? The twist of the bases in a base pair against each other. A base pair is rarely a perfect flat plane with each base in the same lane. Rather than each base has a slightly different roll angle with respect to the another base. This makes the base look like an airplane propeller. What is the pallindrome? Over here there is a figure of a pallindrome. Okay, palindrome are the sequence of double-stranded DNA with two-fold symmetry. Okay, two-fold means it have to be fold like this 2 times. Okay, in order to superimpose one repeat on another, how can we get a palindromic image exactly same as said to be like a palindrome? So we have to, it must be rotated 180 degree about the horizontal. Horizontal means like this. Okay, and then 180 degree about the vertical. First we have to flip it at horizontal, then vertical. Okay, what is the minor repeat? It is a symmetric sequence within each strand. Superimposing one repeat on the another require only a single 180 degree rotation in a vertical axis. Okay, what are the unusual structure that we use to find in the DNA? The first is the bend. You can see how bending is shown. It occurs in a DNA helix where 4 or more. What is the A? A is the adenine residues appear sequentially in a sequence in one strand. Means in one strand you get more than 4 adenine residues. 6 adenine in a row. I hope you know what is the row and the column. So 6 adenine in a row produce a bend about 18 degrees. Okay. The bending observed with this and other sequence may be important in building the same protein to the DNA. Okay. What is the hairpin and what is the cruciform? Palindrome sequences are self-complementary within each strand, therefore have the potential to form a hairpin or cruciform. You can see they can form a bond within each other because there is an AT or TA and GC, and we know purine and pyrimidine, they can form bond with each other. Okay. When only a single DNA or RNA strand is involved in the structure, it is called hairpin. Only one strand when it is participating. When both strands of the duplex are involved, it is called cruciform, like this. It forms a bonding among each other. Mirror repeat do not have a complementary or cruciform sequence within the same strand and cannot form the hairpin and the cruciform. Okay, mirror repeat. They don't have a potential to form the hairpin or the cruciform. Okay. What is the triplex DNA? When the hydrogen bond in the AT and the GC base pair in the B form of the DNA are formed, several hydrogen bond donor and acceptor group in the nucleobases remain unused. Okay. Each purine base have 2 such groups on the edges that are exposed in the major group. The N7, O6 and N6. These are the position. At number 7 you can get the nitrogen. At position number 6 you have an oxygen. And at position number 6 you have a nitrogen of the purine. Participate in the hydrogen bonding of the triplex DNA is referred to as an Hawkstein position. Note. The non-Watson-Crick base pairing is known as the Hawkstein base pairing. You can see adenine, thymine, guanine, cytosine, how they form pairing. Adenine, thymine, at different positions they can interact and form a bond. Okay. The triplex standard DNA requires a homopurine, homopyrimidine region of the DNA. If the third strand is purine rich, out of 2, when the 2 are formed, it is known as the Watson and Crick. But if there is a third strand is formed, okay, if the third strand is purine rich, it forms reverse Hawkstein hydrogen bond in an anti-parallel orientation with purine strand of the Watson and Crick helix. If the third strand is pyrimidine rich, it forms Hawkstein bond in a parallel orientation with Watson and Crick base pairing. What is the Watson and Crick base pairing? It is the AT and the GC. And if suppose there is another strand is present, so always remember, if this third strand is a purine, that is form an anti-parallel orientation, and if the third strand is a pyrimidine, it form an parallel orientation with the Watson and Crick base pairing. Okay. Let's see, the triplex DNA may form an intermolecularity between a duplex target and a third strand. It may form an intramolecularly in a supercoiled DNA. Sorry about the T. Okay. An intramolecular triplex, also shown as a HDNA, could form within a single homopurine, homopyrimidine duplex DNA region. Okay. What is a G-quadraplex or a tetraplex? 4 DNA strands can also pair to form a tetraplex, which occurs in a DNA sequence with a very high proportion of the guanosine residues. It consists of a square arrangement of guanine stabilized by the Hawkstein H bonding. Okay, see the diagram. A monovalent cation is present in the center of a tetrad which stabilizes the tetrad. What is the monovalent cation? This is the, okay. It can form 1, 2 or 4 separate strands of the DNA and RNA. All parallel quadruplex have all guanine glycosidic angle in an anti-conformation. Always remember this statement. Anti-parallel quadruplex have both sync and anti-conformation. Okay. Let's see the stability of the double helical structure of the DNA. The first is the hydrogen bonding. These are the hydrogen bond. I hope you are able to see. that form between the complementary bases, that is the AT and the GC. What is the double bond? That is the hydrogen bond. Okay. Hydrogen bond form externally between the polar atom in a sugar phosphate backbone and surrounding water molecule. Okay. Then base pair stacking interaction. What do you mean by the stack? Stacking mean, have you seen the when we put the coin one above the other? It is exactly the same. Okay. It includes the hydrophobic and the van der Waals interaction between the base pair. The base stacking helps to minimize the contact of bases with the water. The negatively charged phosphate group is situated on the exterior surface of the helix in a way that they have a minimal effect on each other and free to interaction electrostatically with the cation in solutions such as the magnesium. These are the negatively charged phosphate group, okay, and interact with the water. Thank you so much.